Good afternoon or evening to all of you, depending on where you are, and thanks for joining us today for our annual update on research matters in PXE International. This year's webinar is called PXE Research Planning in 2015, and Patrick Terry will be presenting all the new results and where we're going with research on PXE during this year. Patrick is a dad of 2 children with PXE who are now young adults. He's also the co-founder of PXE International with his wife Sharon Terry. He's the president of the board of directors of PXE International, and he is a recognized leader in genetics and health policy and research questions in the community. As I was probably muted, I want to review the housekeeping issue, concerns. If you have any questions, you can type them into the question pane. You'll find the question pane on the right side of your screen in the dashboard there. Type them in, I'll receive them, and I'll present your questions to Patrick. We do welcome your questions, so please put anything in there that you have. If you have any technical issues as well, enter those in the question pane and we'll see them, and we'll respond to you personally in the question pane if it's a technical question with your own sound or whatever. So with that, I'd like to turn it over to Patrick Terry, and thank you very much for being here.
Thanks, Chris. Thanks, Terry. Thanks, everybody, for joining. So as Chris stated, I'm going to cover research planning. So this is the perspective from PXE International, the organization, and the board of directors. And I want to talk about plans and priorities, or focus areas, if you will, and then share with you next steps, things out ahead of us that we want to plan and program for. So with that, I'll start the presentation, and then please feel free to type a question, and then I'll get to that as best I can. So just as an alert, so there's the PXE International Research and Update and Anniversary meeting. This is actually a bittersweet event for me personally. I had hopes and expectations of not requiring such an anniversary, but here we are. So 20 years ago, we started PXE International, and for the aspect or the response to having 2 children diagnosed with pseudoxanthoma elasticum, and then quickly learning and meeting individuals that have been living with this disease and coping with it for a long, long time. And my 2 children grew into 2,300 children early on in the foundation's history. Now well over thousands upon thousands of individuals that we care for in one way or another, or aim to help through the foundation. And I think part of the blessing of that experience has been expanding my universe, my horizons, my interaction with people I would have never otherwise have met, as well as interact with some fantastic individuals in the research and clinical community that we not only consider colleagues, but close friends. So this October, we're going to be celebrating 20 years of research in progress. We'll be discussing scientific research and medical care updates and recent findings, some clinical trial updates, as well as some information from the PXE Help Survey, which I'll talk a little bit about here. But just as a reminder to put this on your calendar, it should be not only informative, but a fun time. So research planning, for today's presentation, I want to talk about what our priorities are, what we're doing in the aspects of work with our collaborators around the world, and some advances of understanding the underlying causes of pseudoxanthoma elasticum, the impact on the body and the systems, as well as the various experimental models, both in vitro and cellular and animal models and intervention studies, treatment paradigms, and some human clinical translational research that we're actively involved in. And as any of you know that have been following PXE International, we embarked on a heavy focus on research the first year of our founding, providing all of our research funds to Jackson Laboratories to screen for a wild-type occurrence of Pseudoxanthoma elasticum in the mouse population in that animal laboratory. And it's obviously now since expanded to 19 different research groups around the world, and helping train students and investigators on this disease, and now being an integral component and contributor to the research and clinical enterprise that undergirds what the foundation does today. So I think also I want to highlight that there's some areas I won't be covering today because they, Chris and Terry have effectively put together a webinar series with a variety of researchers and investigators that are working on PXE, which they've talked about their own work in very eloquent ways and in detail that is not only accessible to the layperson and to our general audience, but is really informative on their perspective and their research on both the ABCC6 gene or the MRP6 gene and the protein and some of the human disease consequences. So there's Doris from Germany and Koen from the Netherlands. Sharon presented an epidemiological and health research study that we've asked participants to join. And then the last and most recent webinar from John was a very good one around some of the new learnings around PXE. So I'll just refer you to those. Those are on the website. Those are archived, and you can watch them at your convenience. I thought it was important also to maybe just highlight myself and my background a little bit. So when my two children were diagnosed with a disease, PXE, and the disease diagnosis from Lionel Berkovich, who is now our medical director of the foundation, 20 some odd years ago, I was just a dad. I was a construction manager in Boston, Massachusetts, and wanted to make a difference in their lives, so I got involved, and we started this foundation in 1994 because of Elizabeth and Ian, and my wife, who was a campus minister and a theology major, also got involved with running a research foundation, and we knew nothing about human research, genetics, any of that. So this is really an enterprise of love and learning. Based on that experience and that my work with PXE International, I left the construction industry. I've started a number of for-profit life science companies in various disease areas. I've been lucky enough to be very successful in raising money and changing the disease pathways for breast cancer, lung cancer, prostate cancer, infectious disease, a variety of other disease states beyond the rare disease of PXE, but it has enhanced my capabilities and my knowledge and my network in this space to be truly successful in helping PXE advance its objectives. And I think, you know, I'm about 3 weeks into a new position, which is quite exciting because 20 years ago, my two children were diagnosed at Boston Children's Hospital by their geneticists, and we went through that process. And again, Lionel Berkovich made the initial diagnosis for them both. And now Boston Children's Hospital has hired me back after 20 years to run their genetics and genomics program and their commercialization effort to bring genetics to rare diseases and pediatric diseases of all sorts, including PXE. So that's it's a full circle for me, and it's quite exciting. After the work with PXE International, I started Genomic Health, which is the cancer genomics company that went public and is still in operations today and doing quite well. I started the Personalized Medicine Coalition and the European Personalized Medicine Coalition, 21st Century Medicine Coalition, the International Genetic Alliance. So a variety of umbrella groups that are working on policy, federal efforts, international efforts to advance applied genetics, human research, rare disease research, and so forth. So I've expanded my influence and my network around the globe to the point now that we have honorary professorships at universities and giving lectures to MD and PhD students on directing their careers into helping patients with rare diseases. And it's interesting now when you look back at not knowing anything about genetics and medicine and healthcare, that between my wife and I, Sharon and I, we have, I think, over 60 or 70 peer-reviewed scientific publications and high-impact journals. So it's pretty funny to go from just a dad to being an influencer on this stage. So the foundation was organized around advancing research. The project plan in which I put together when the kids were diagnosed, I'll talk about specifically. But I think now in the maturity of 20 years, what the foundation is really most urgently focused on is to taking advantage of any opportunity to alleviate the burden of disease, and that's finding treatments and solutions for patients that are living with the disease manifestations today, as well as looking forward and working on developing interventions and treatments and knowledge and advancing clinical understanding of the disease in the long term. And in essence, what we are attempting to do as an organization is to tame the PXE disease process. I think it's a little bit hubris to think that we're going to be curing disease, but if we can stall the disease process, mitigate some of its consequences, delay onset or severity of this disease, so we're just dealing with issues of normal aging, that would be a success as far as I'm concerned. And those are the kind of the two focuses or lenses, if you will, on how we look at what we're trying to achieve with research. Our research priorities, they're not necessarily in this order, but it was a list here that I tried to imagine is somewhat interconnected or sequential. We have basic mechanisms of the disease process, and we've been working on that for a good 20 years. It's starting with the genetic underpinnings of the disease. How does genetics result in the pathway to create proteins? How do those proteins interact with the cell and causing a cell to function or not function? How does that cell operate and function within a tissue? How that tissue functions in an organ? How that organ functions in the body? And then how, from a holistic point of view, how does an environment interact with that organ and that human system as it relates to PXE? And why is that happening? Why has it been conserved in nature all these many years through all of these different species and organisms? There must be some type of selective pressure or advantageous outcome or derivative from this mutation in either a lower life form or in humans, and what might that be? So basically building a narrative to understand what are the mechanisms here from a conceptual standpoint. Then we want to understand the PXE pathogenesis. So pathogenesis is a fancy word of saying why is something happening, why does it manifest the way it does, and how does it impact human biology, disease and health, if you will, and understanding that from a clinical as well as a medical perspective is important. Understanding this aspect of metabolic pathways, since this loss of function in this gene, when this gene is mutated, the protein isn't functioning. It's a loss of function. It's a loss of capability within a certain cellular system in an organ, mostly the liver in this circumstance of PXE. And because of its liver function impact, it's described as a metabolic disorder. And understanding how that is, why that is there an enzymatic effect? Is there a substrate or a molecule that isn't or is in circulation that can be manipulated or enhanced in a metabolic approach? In a certain aspect, it's actually quite amenable to therapeutic approaches or enzyme or protein replacement therapy if it is a metabolic disease, if you have a loss of function because this molecule is in circulation in the blood. It's obviously quite easy then to deliver a new molecule or a treatment systemically through the blood through an infusion. We don't know if that will actually be the avenue of an effective treatment or therapy, but it is certainly a possibility and a likelihood based on a variety of experimental discoveries and animal studies that have been conducted that I'll talk a little bit about later on. I think also the tissue and organ processes are important. We have this ongoing debate whether this is a loss of function in the liver, or indeed it's a multiple impact on different cells and different tissues in the body that result in PXE. They're not necessarily mutually exclusive. They both could be happening and contributing to the severity and process of disease. So again, understanding not only what's taking place and happening in the skin, what's taking place in the vascular system or in the lungs, in the brain, in other tissues that are not impacted by PXE, and understanding why those processes are occurring and why some organ systems seem to be protected or immune to mineralization, while other organs and tissues seem to be quite readily impacted and severely impacted by PXE. I think once we understand some of those mechanisms and processes, then this is spelled wrong, but the phenotypic causality and prototypical causality, so we can understand what is the typical process, what is the typical phenotype, what is the time to progression, what is the likely severity of disease, how does environment or other genetic components or contributors impact the disease, and making sense out of a more complicated system of interaction. Obviously, diet, exercise, lifestyle have clearly demonstrated that they have a large contributory role in the disease and the disease severity. By way of example, we've seen that smoking contributes quite heavily to some of the complications associated with PXE, and making sense out of the interaction effect of some of these environmental and lifestyle factors, and understanding whether exercise and diet or mineral intake and other supplements can impact the disease process, and really ferreting out in a rigorous way what helps, what hurts, what can improve outcome. Therapeutic approaches are important because we believe that there's going to be a variety of not only drug therapy approaches, enzyme therapy, there could even be other organ and system manipulation. How does stem cell and regenerative medicine play a role here? So we both have approaches and considerations for a pharmaceutical compound, repurposing existing therapeutics or existing approaches to impact the pathway and impact the severity of disease, doing human clinical intervention studies, doing animal model studies with some of these therapeutics, potentially even working and doing a number of compounds in development for drug screening. So a whole new drug development process that could be applied to PXE. PXE is what's called a prototypical disease of mineralization, so there's interest in other disease areas that have an overlap of mineralization that aren't necessarily associated with PXE, that may be further along in therapeutic development, or may be quite interested in being involved with our therapeutic development because of discoveries or influences that we can demonstrate with PXE that could have relevance to a normal aging process or advanced aging processes in other diseases. So we certainly try not to think of PXE in isolation, but think of it broadly and holistically to, again, take advantage of every opportunity to alleviate the burden of the disease. So our process, simply described, is we want to translate and work closely with academic research findings. We want to support clear and high-quality evidence generation. And we want to do this in such a manner that we have multiple experiments that verify results. We want to confirm those results in tertiary centers. We've all experienced kind of the hype of a new discovery published in the literature or published by PXE International, and then individuals run with that data or that early information with a vitamin K finding or a magnesium finding. So we're also quite mindful and want to be cautious about these initial findings because we've demonstrated again and again that they do not hold up under rigorous analysis, and patients shouldn't be messing with their vitamin K and magnesium and calcium levels based on some of these experimental findings. We want to really confirm these results. We want to do in silico, basically computer analysis using in vitro studies, which are experiments in petri dishes, and then functional assays or disease models in cellular systems to test these hypotheses. Ultimately, we have a number of animal models. Animal models are important, but they're challenging because while we have phenotype of high fidelity with our animal models, meaning that PXE in the various murine or mice models mirror similar manifestations to the human disease, but ultimately when you start manipulating those systems with either drugs or approaches or dietary regimes, that information and that data again and again has shown that it does not necessarily completely translate to the human. So while animals and mice and zebrafish are important, ultimately, if you can cure disease in a mouse, does not mean you can cure that same disease in a human. So we have to be cautious there as well. So we only want to move on to human experimentation when it makes clear, safe, when it looks like it's safe and effective. If there's an existing therapeutic or an approach that can do no harm, that may be beneficial, those things we will pursue. I think we would prefer and obviously want to be doing controlled clinical trials, but in some circumstances that is not proven possible or feasible. Ultimately, the example here would be the anti-angiogenic compounds, or these compounds that are attacking new blood vessel growth, and this is important for macular degeneration in the experience that PXE patients have with breaks in the Brooks membrane and hemorrhaging in the eye. These therapeutics that have been developed for cancer, because cancer also produces new blood vessels, and now these cancer therapeutics have been demonstrated to be effective in age-related wet type macular degeneration, which is a disease or a disorder of the aging process in tens of millions of individuals in the United States, that those therapeutics have proven effective in PXE, which has been a wonderful occurrence, because rather than having patients call the foundation every day with losing vision, where that's actually a rare occurrence now. So these injections of these anti-angiogenic compounds in the eye have been effective for treating the symptoms of the ocular manifestations of PXE. It's certainly not a cure. It doesn't solve the problem, but it has prevented further vision loss in most patients. So that has been terrific to see. But we also have a discipline and a focus of wanting to refuse to support activities that are not rigorous or not ethical from a variety of perspectives. And this year has been an example of multiple clinical trials and human therapeutic approaches or intervention approaches that we have determined as a foundation are not ethical or not safe or not proven. And we don't endorse them, we don't participate in them, and to the extent that we are able, we try to counsel and influence those groups not to subject patients to these potential harms. So I think that will be something that we will continue to have to manage, and it's not an easy thing for the foundation to do. We want to support research and promote advancement, but not at any cost. So I think these discoveries also need to be accessible, just because it can be done, and it's been demonstrated in a handful of patients that if the data is not rigorous, if it's not controlled, then there's no way you can convince a third-party payer or fiduciary agent like a national health policy or Health Technology Review Committee to cover this and to pay for it for patients. So unless these things are covered and integrated into routine care, then ultimately patients won't have access. And we see this again and again, even with the angiogenic compounds that are very effective here in the United States and Europe. There are Eastern European countries and other places that they still cannot gain access to these very effective therapeutics. So there's a lot to be done there, and the patient group and PXE International can be an agent to make sure that patients have access. And we want to make sure that our clinical collaborators and academic collaborators kind of do it once and do it right, because we can't waste time for doing it quick and dirty and then having to do it all over again so patients can have access to these results. So the project plan that we put together 20 years ago, the rationale was, let's understand the genetics, inheritance, and get to a therapeutic intervention as quickly as possible. There's these various domains of science or task-based activities or disciplines in both the discovery and translational and clinical enterprise, and I've learned a lot about these domains when I wrote this map 20 years ago. It basically hasn't changed since then. I knew these terms. I conceptually knew really what the domain of activity was and potentially what the interaction or feedback system may look like from one domain to the other, but I didn't really have a complete concept of what this means. Now today I know both the academic, industry, federal, international enterprises that are working on these various activities, and the researchers and the technologies and the and even the financial aspect of the transaction and the research funding that makes all of this work. I think a few years ago we did a research update, and we figured that about 80% of this activity map PXE International and our collaborators are working on. So that's highly satisfying that we've mapped a systematic approach to disease discovery, and 20 years later there's activity still happening here. I think in hindsight or post hoc analysis here, I wish it hadn't taken us 20 years. We've seen examples now where we have mentored other disease groups to try to model what we've done, and some groups have been tremendously successful both in shortening the time for some of these discoveries and advances, and even most recently with the Cystic Fibrosis Foundation actually developing highly effective compounds and therapies. And because the patient group was involved with all of this drug development process and these therapeutics, they actually just sold their shares or their stake in a company, and $3 billion went back to the Foundation for Cystic Fibrosis Research. So you've seen some fabulous examples of groups that have been really effective in their own disease areas. So what have we done? We've created a biobank, a biorepository that we continually use. So this is a contact database, biological materials, medical records, longitudinal history and data around patients. We're involved with the gene discovery. We're the only lay group and patient group to patent the genes and then manage the stewardship of those genes to produce diagnostics and therapeutics and manage the in preventing price gouging and profiteering in this domain of translational research and product development. We've created diagnostic tests. We've been involved with organizing human clinical studies, and now certainly embarking on drug development in therapeutic approaches for PXE. And our aim here is obviously to find a solution and to tame the disease. And we're using all the tools and avenues that are available to us, including contract law and business frameworks for us to be successful, to make sure that the patient is at the center of this enterprise, and that we're staying focused and staying tempered and urgent-based in how we make some progress here. This is just a quick little map of, we have research collaborations and research groups, both clinical and basic science, around the world. And these individuals are professionals in their domain, whether they're in academia or in industry. And this has been one of the most satisfying aspects of PXE International for me, is to both learn science and the conduct of academic research and discovery work that these individuals do professionally, and to participate and to support these activities, and watching individuals grow, learn, increase their professional responsibilities, and then become chairmen of departments and running their own groups. So from a student to now a primary investigator over these past 20 years, that's been really interesting to see how that has happened and how we've contributed to some individuals' professional success. I think another aspect of what we do and what we want to do is support information transfer and interaction. So we facilitate and support meetings. Most of these meetings are biannual in Europe and the United States. So we participate and underwrite and support student travel, research dollars for abstract and poster presentations and podium presentations around pseudoxanthoma elasticum and the other transport proteins that we're involved in. So in essence, now the foundation has been involved with supporting research work on 16 other proteins that are related to ABCC6, hoping that that work will inform our understanding of PXE as well. So we're interacting with a much broader community of basic researchers, and not just those associated with PXE. If we do a tally of some of our achievements and what we're doing activity-wise, here's just a list of, I've already kind of talked about all of these things to date, but it's pretty fantastic to think of a lay, patient-driven group having so many activities that have driven science and discovery and knowledge forward around PXE and around related diseases. And in essence, here's a drawing we did back in 2000, just after the gene was discovered, and we found out that it was a membrane transporter. So this cartoon here is a slice, if you will, of a cell, and this protein is a complicated transmembrane protein, and it creates a channel or a pathway between a membrane, and it is an active efflux or an active pump. It's a very conserved and complex protein. It's essential to biological function in every living organism on the planet, and it has different roles and functions, which has been somewhat frustrating because it is so conserved and it is such a complex structure that it does a lot of things. So trying to figure out exactly what it's doing, what this molecule is being transported by ABCC6 has been difficult to isolate, because in certain experiments you can set it up in trying to isolate what that one thing is, and we've discovered that it's what they call promiscuous. It will pass and transport a lot of different things, and trying to figure out if those lot of different things, any one of those are related to the PXE manifestations or not, has been the real challenge over the last few years. Here's just a cartoon again of this two-piston pump mechanism. So the protein has this catalytic effect, and it needs this energy to move molecules from inside the cell to outside the cell, and when that's interfered with or missing, then we have the manifestations of PXE. This is basically a simple map of what the protein is doing, but how it's doing it and with what molecules is still under determination. The work out of Yoni Wido's group in Jefferson University in Philadelphia, they did some elegant demonstration in animal models around demonstrating that PXE is a metabolic disorder or a liver disease, and that work has been confirmed by a number of groups, and there's a number of animal models out there, both similar between research groups and as well as independent models and different models of mouse models to show a similar scenario where this missing ABCC6 gene in the liver and pumping something into the blood and circulation is responsible for the disorder. Now we're trying to figure out what those pathways are and what's missing and why and what's the impact. Adenosine and ATP transport in the liver or out of the liver it still needs to be elucidated a little bit further, and there's a number of groups working on this now to determine really what is this function. Is it completely an hepatocyte or liver operation and transfer through multiple genetic pathways or protein pathways, or is it this interface at the liver, the hepatocyte, and blood circulation, and a systemic effect? So all of those models are being looked at and studied by a number of groups. I've mentioned the mouse models, and these mouse models have been really important to understand not only the genetics of PXE, but the phenotype. And in essence, what happens is the mice have this prolific whiskering at the nose, and it's called vibrissae. And at the vibrissae, or the hair follicle root on the snout, it calcifies quite readily when these mice have PXE, the human PXE gene, inserted into their genome. So in essence, they mimic a similar calcification of the fibroblasts of the skin and the cardiovascular system, and they have, if you age the mice out as elderly mice or post-breeding mice, you can see typical PXE, human-like PXE manifestations in these rodents. These rodents have been also important in these model systems for understanding can this loss of function be rescued with different therapeutics, and Andras Verity and his group in Hungary have demonstrated with different types of mutations in the PXE gene that they can use certain therapeutics to rescue the protein so it doesn't get degraded with RNA or DNA degradation within the cell, and so it'll transport through the ER and the Golgi apparatus, and it will go to the proper location in the cellular membrane, and it will have partial function. And I think the other aspect of this data that was really compelling is to show even if you gained partial function in the ABCC6 gene, the phenotype was potentially alleviated. So if you could upregulate function by 5 or 15% in these model systems, you could actually prevent the manifestation of mineralization or calcification, which is quite exciting because then you don't necessarily need a perfect therapeutic to interfere with the PXE process. You could have an imperfect or partially functioning or a mild influence of a therapeutic on PXE and still have a dramatic impact on mineralization of the soft tissue. So that was an exciting aspect of this work and opens up a lot of avenues of potential approaches or therapeutic endpoints, if you will, for clinical study designs, both in animals and in humans. Another important piece of work, again, the excellent and elegant research done by Yoni Wido and a number of his postdoctoral fellows in their research group at Thomas Jefferson University in Philadelphia, and it's the zebrafish. And this is an interesting model because it's completely transparent from the single egg form all the way into adulthood. So looking at and seeing and using it as an observational animal model, they not only grow very fast, but you can see what happens with the vascular system and the cardiovascular system and even the neurological system of this animal model because it's see-through. Some of this work has been also important to demonstrate that the PXE model in the zebrafish is either lethality or what's called a curly tail phenotype. So rather than mineralization and calcification, they actually get curly tails. And again, the Wido group has been able to show with Andras Verity in Hungary, in Budapest, that some of these therapeutic approaches could actually rescue the curly tail phenomenon. So again, you're preventing a PXE manifestation in zebrafish. It's just another demonstration that these drugs and these approaches do indeed change the biological consequences of loss of function in ABCC6. Doing a little transition now into human clinical studies. So the We've spoken about and supported the Mount Sinai clinical trial. So this is work that was informed by Yoni Wido and his group with the animal model, showing that dietary intake of magnesium could influence the severity of disease, and we've have an FDA-approved protocol of a dietary approach and research happening under a controlled human study out Mount Sinai. We've worked internationally to help develop research centers and collaborations around the world so we can understand different patient populations around the world, and one of the most important ones, again, that the Wido group has been highly influential in arranging is establishing interactions with the Chinese government and health systems and military hospital systems for a PXE Research Institute in looking at the Han Chinese and discovering different mutational profiles, different clinical manifestations due to PXE. So this has been an important collaboration that we hope to expand upon and to model forward in other perhaps ethnic continent of origin populations, perhaps that have different manifestations or different disease phenotype associated with PXE that we would need to study and understand. Another group that in recent interactions we've been highly interactive and supportive of, and this is the group Piet Borst in the Netherlands. He is a chair emeritus of the Cancer Institute there, and again the MRP or multi-drug resistant proteins that ABCC6 is a part of. There's been a tremendous amount of work and study and research done on these proteins and these genes as it relates to cancer. So the Cancer Institute is highly knowledgeable about these proteins from various perspectives, mostly from the cancer perspective, but now that PXE International has been supporting research and animal studies at this institute and working closely with this metabolic approach, they've made some seminal contributions to both the knowledge and understanding of PXE and doing metabolic screening, and have advanced our understanding to the current point of this ATP and adenosine and pyrophosphate interactions related to PXE, and helping to advance the community, their understanding and the community's understanding, and helping to drive a research agenda for the foundation. And meanwhile, while all this research is happening, the foundation also wants to stay focused on lifestyle and exercise, and we've done a variety of efforts, and now Sharon is spearheading this epidemiological study around behaviors and lifestyle, since we want to capture this information and understand it, so we could potentially make suggestions and recommendations to the community, that things they can easily do themselves to help live better and healthier lifestyles and influence the disease process, so they are in kind of control of their own destiny. So this involves dietary regimes and approaches and supplements, and this highly interactive dynamic between calcium, magnesium, and phosphate in the body, and how to regulate or not regulate this effectively, and how individuals can change some of these concentrations in their own body by diet and supplements, but also we want to educate the community about the the horrendous consequences of calcium restriction and doing things in the extreme that can actually do more harm than good. Again, John Schoenaker's presentation around calcium and magnesium and pyrophosphate and phosphate levels and hydroxyapatite and mineralization of soft tissue is an important webinar for everyone to understand and to listen to. So we will continue to support and interact with our close collaborators in Hungary and the Thomas Jefferson University and the broader research community, and John now at Emory, and advance this research agenda that is around basic science, animal models, mechanisms, and therapies, and driving some consensus among the scientific community on how to prioritize some of these things and how to set certain milestones so we know we're making progress in a rational way, and that we're taking advantage of facts and data as effectively as we can, and share this information and this discovery work as quickly as possible so the community can collectively make efficient progress in a timely manner. So some of our approaches have been a systematic metabolomics approach that proved some unsuccessful in the mouse, some transcriptomics work, some transfecting cells. So we have some dog liver cells and some human cells. That work is still ongoing. Some substrate experimentation, some of the vitamin K work. A lot of these approaches and these experiments have resulted in negative findings or unsuccessful findings as it relates to PXE, but it also defines a road and an exploration in which we've attempted, and we should move on to another hypothesis and another approach, because we've rigorously done an analysis, and the vitamin K is an example of that, of multiple groups, lots of effort, and then collectively coming to the conclusion that that's not related, and that hypothesis was wrong or incorrect. Also working around key questions and interacting with the research community and the clinical community around what these key questions are, what would be the most effective experiments to answer these questions, what order should we do them in, should we divide and conquer, should we agree that one group should do this and another group do another thing, and working with the groups to determine about who's not at the table, who's not here, what expertise do we need, what kind of collaborators should we go search for. That's an important aspect of expanding the knowledge and our experience by dealing with groups and investigators that traditionally have had nothing to do with ABCC6 transporter functions or PXE specifically, and how to engage with them and get them interested in contributing. I also want to highlight that PXE and the staff do an excellent job of mining the literature, gaining access to these medical papers that sometimes are under subscription-only access, and doing research summaries and reports and lay reviews of the basic science and discovery and putting it on our website. And you can find it by just going to our website and looking under events and resources and finding those things. You can view them in large text format on the website or print them out for reading if you prefer paper-based, but it's a terrific repository and hub for staying current. We also do a newsletter that tries to keep the community abreast of what we're doing and how we're doing it and when things will happen and how we'll report them to the community. And you know, where we're going now and the next steps, and this is where I'll conclude, is this aspect of therapeutic or intervention approaches. So we're going to be interacting with new and different types of investigators, clinical trialists, experimentalists, interacting with regulators. We have already had a number of challenging interactions with the FDA around repurposing the angiogenic compounds, getting an indication for use in PXE as an example. I think getting these molecules or a number of molecules under study for PXE rather than making a bet on a single drug and a single molecule. We are talking to the regulators to do a group approach where we would bring forth multiple therapeutics into a single regime and a single submission for clearance and approval. Also working with the regulators to consider the best pathway forward for using or repurposing existing and approved therapeutics for PXE. As we've gained some insight now into potentially what's happening or not happening in the liver, then dealing with industry, both biotechnology and the pharmaceutical industry, about who has compounds, who's working against pyrophosphate in the body, or working for therapeutics that are effectively managing osteoporosis in adults, and whether those therapeutics could be made available for experimentation with PXE, those types of things, and dealing with industry for both sponsorship and access to compounds, because ultimately I think now we're going to be turning the corner into a much more expensive and complex research enterprise, because we're going to be getting into human clinical trials, therapeutic studies, industry, and a very complex process of getting therapeutics approved and available to patients. So with that, that's my conclusion, and that's a real high-level view about our research plans and priorities.
Thank you, Pat. We do have a few questions. You mentioned rescuing the zebrafish. Can you expand on what that means, and does it mean that a zebrafish that has the curly tail can be rescued to not have the curly tail?
Yeah, so both the so rescue is a fuzzy word. So it's in the experiment itself, you have zebrafish that have this curly tail phenotype. So they have, rather than a gene inserted or a knockout, there's a different experimental technique called it's a morpholino, it's called, and it's called a knockdown. So basically you chemically manipulate the zebrafish's genome to find out, so we know where the ABCC6 gene is on the zebrafish, and if you knock that down, so you cause it to lose function at the earliest stages of development, those fish develop curly tails. And if you take these trafficking therapeutics and you put it in the water of these zebrafish, and they infuse the water and process it through the zebrafish itself, those curly tails go away. So basically the fish will then develop a normal extended linear tail. So the rescue is basically rescuing a phenotype. It's not necessarily curing the fish of PXE, but it's just demonstrating that these compounds or these therapeutics have a direct interaction with the phenotype that's caused by ABCC6. The same has been demonstrated in the mouse cells and the dog liver cells, that there is interaction effect. That so you have a phenotypic sign or symptom caused by PXE, and if you expose these cells or these animal models to the therapeutic, you can correct the phenotype, or in my slides I talked about it as a rescue of the phenotype.
Mm-hmm. Good, thank you. We have another good question. What can the community of people with PXE and their families do to speed the progress of research and help PXE International with that?
Yeah, well, it's not sexy, and it can be fairly burdensome, but participating in the survey and the PXE International survey instrument, I think contributing your data so we understand most rare diseases have some discovery, have a potential therapeutic, and they bring that to the FDA and they say we want to do experiments and we want to do a human trial and we want to get this drug approved because we've demonstrated in a handful of patients or one patient that we can cure them. And the FDA says, well, where is the phenotypic understanding? Where is the biomarkers? What's the progression of this disease? Can you demonstrate therapeutic interaction effect? So they ask all of these regulatory type questions that the sponsor or the investigator doesn't have any data for. So then they have to go back and collect patient populations, collect epidemiological data, develop a biomarker, and all of that delays this process by years and years. So if we had a biomarker, if we had all the patient population, if we had the longitudinal data, and we have that characterized in a rigorous way that we can submit in a regulatory package to the FDA, we're way ahead of the game. So we're slicing years and years off of development time. So that's important. So while PXE International may look like it's just wanting your data, there's a reason why we want your data. There's a reason why we've done the epidemiological survey at these various time points over the last 20 years, so we can demonstrate exactly in a handful of representative patient population how the disease progresses, how mineralization occurs in the various components of skin and the arterial systems and the eyes and so forth, because that's important for regulatory clearance and approval of a drug. I think also, we obviously ask the patient population way too often for money and for financial support, but ultimately, any contribution in that regard, even if it's modest, is extremely important, because to keep this machine going and this apparatus moving forward, it takes money and resources.
Yep. Thank you. We have a couple more sort of definition things. What did you mean when you said the gene was conserved? I think you said that about in zebrafish. Or the gene or the protein was conserved.
Yeah, so we did some really interesting work, what was called comparative genomics. So if you take a significant portion of the genetics, so the DNA that make up this gene, that make up this protein, are conserved, meaning that in the lowest forms of single-cell organisms and yeast and plants, that this gene is as old as life itself on this planet. It's a basic building block component of multicellular function in both plants and animals. And so that's what I meant by highly conserved, that you could follow significant components of this gene and protein all the way back through evolution and history of life on this planet. So my other comment about why this gene has been conserved and why these mutations have been conserved all the way to humans in the evolutionary tree is perhaps in a lower organism or life form or in plants or something, that these mutations had survival advantage, and then that they were just conserved all the way to the human genome. So we did some interesting work with some collaborators at NIH where we did computer analysis, and you could take 50 or 200 different animals and plants and overlay their genomes, and you could view exactly where this gene is, and it exists in different locations in a chicken genome or a banana or a mustard plant or zebrafish in a mouse. But the gene itself is highly conserved, that these components of this protein exist in all of these different species.
Thank you. That's a good explanation. There's a couple questions about clinical trial. The one in Mount Sinai on magnesium, when might we have results? And I think a related question is there a biomarker or something you can measure to determine the severity of PXE, and are they using that in that trial?
Yeah, we do not have a good biomarker. We will probably in the near term here have a subcellular or a tissue biomarker once we completely understand what the pathomechanism is and what's happening in the liver and these hepatocytes. There could be a metabolic or circulatory biomarker. So the probability that we will have an excellent biomarker for PXE for our therapeutic approaches is I'm highly confident that we will have one and have a good one. We don't have one today. Right now what we're using is observational phenotype, so severity of plaque or semi-quantification of mineralization in a fibroblast or skin, or a morphogenic analysis of mineralization of the Brooks membrane in the eye, but these are all highly subjective instrument-based observations of various acuities. So they're not great biomarkers, but we will get better ones, and we're working on getting better ones. And having that biomarker, and if it indeed correlates with some of these observational aspects, as well as our epidemiological data that PXE International has been collecting now for 20 years, then it makes it a rock-solid biomarker, and the regulators will have confidence in it. So all we have to show is interaction effect rather than cure or amelioration of mineralization of the soft tissue, because the data will be so sound and convincing that we could possibly submit an accelerated approval process through the regulators. And this is exciting, because if we have this circulatory biomarker, if we have an existing and approved therapeutic, we would just need to do a small clinical study, show interaction effect to this biomarker in this therapeutic approach, get a label indication for that drug for PXE, get FDA approval. We would be off to the races in a 3-year time rather than a 10-year time for a therapeutic approach. So we are working really hard and realize the power of having good biomarkers, and if it aligns with all the data that we've been previously collecting, we'll be in a good spot.
Great. I like to hear that confidence that we're going to make it. Someone asked, what age, how early can a baby, a young person, be diagnosed with PXE?
Well, it is an inherited disorder. So if you look hard enough, you could find it. So you could probably even find it in gestation. So it's there. It's a slow progressing adult onset condition. So the causality of the disease is there from prior to birth, but the phenotype and the consequences of the disease obviously doesn't become manifest until later in life or over time. So the earliest, maybe we could find a circulating biomarker, and from that biomarker you could do an amniocentesis and discover that the baby you're carrying has pseudoxanthoma elasticum, and the best case we could, this is going way out in the distance and speculation, but then offer that therapeutic to the mother herself, and while in gestation you could actually be treating the baby for pseudoxanthoma elasticum. So there are all kinds of scenarios we could think of, but indeed the genetics here are quite clear that you're born with this condition, and maybe we can do a variety of different manipulations to impact the disease progression and make sure that anybody born 10 years from now is not worrying about PXE and skin lesions and eye lesions because we have an effective preventative approach.
And so you mean that what could be done now is potentially a genetic test with using a blood or saliva to just search for the genes? Is that what you mean?
Yeah, you'd have some signature that gene is there, and this is going to be true for all the genes in our body. That ultimately newborn screening, 25 years from now, every baby, 2 days post-birth will have a newborn screening test and test their entire genome and understand the 40 or 50 genomic insults that every individual is born with, and understand that there's a handful of clearly preventative actions that can be taken. All kinds of adult onset conditions or developmental conditions can be treated and alleviated days after birth, and that's going to be a profound implementation of genomic medicine for everybody that's born around the world.
Yeah. One question that I think you missed was, do you know when the human clinical trial at Mount Sinai will be giving us results?
My expectation is we will have them this year. Every update that the Mount Sinai investigators and group have presented have been dissatisfying because they say we can't disclose results yet. The data has been still masked. So I don't know the exact date, but it should be happening in 2015.
Great. I think they're the trial is going to conclude in April, but what's the word on how long it would take to get it analyzed and published? Do you have an idea of that?
Yeah, normally that takes a few months, and then a data review board, and then they would want to get it published, presented at a meeting and then published. So all of that, could take to 3, 6, 9 months once the data is actually produced and finalized and ready for analysis. So it's a lengthy process. My expectation is it will be mildly informative. It was a small cohort of only 44 individuals, so it will be maybe another link in the chain here that, again, there's a delicate balance between magnesium, calcium, and phosphate in the body that can influence disease progression. But again, their endpoints or their biomarkers are looking at skin lesions and the probability that you're going to be able to eliminate or rescue affected skin with magnesium intake is quite improbable. So I don't hold a high degree of hope that this is going to be a profound study or a profound result.
Mm-hmm. Thank you. There was one more question. At this point, we were talking about potentially diagnosing a baby with a genetic test. Would that kind of testing be covered by insurance?
Well, in the United States, newborn screening is covered. What's included in the newborn screening is all over the charts. So if you're in Mississippi, you get tested for 8 things. If you're in New York, you get tested for 64. In Massachusetts, 80-something. So all of that has to change and evolve because all of these, in the next few years, all the genetic diseases for the 3,800 human diseases that have been characterized and associated with a gene will be available. So states will have to figure out how to deal with that, and payers will have to figure out how to deal with that, and costs are dropping really quickly, so there will be companies and service vendors that will be able to do this cheaply, and even more cheaply than the states are doing the 4 to 80 genes. So I think it's inevitable that this type of testing will be available, and it will be paid for or subsidized in some public way.
Well, thank you very much, Pat. We enjoyed the update. Thank you for taking the time to inform us today. We don't have any more questions, and so I thank all the participants for coming. We have one last bit of information to show you in a second. And I think Terry needs to put up a slide. Basically, I want to remind you once again to fill out the survey if you are willing. It will really help us get ready for any treatment that might come along that we could test. You can find the survey right on our website at that link that you see on the screen. You start it right from there, and you don't need to go anywhere else. It's all online. And if you need any help, we are here to help you. Please just email me, cvokey@pxe.org, or info@pxe.org. The whole staff is ready to help you get through any difficulties you have. If you have low vision, we can help you go through the survey, even on the phone. If you have any glitches in the survey, just get in contact with us. Also, save the date for the PXE International 20th Anniversary Conference, October 16th to 18th, 2015. It's in the DC area where we're located. And I wanted to announce that we have a date for a second webinar by Dr. Jonathan Schonecker to tell us more about calcium and phosphate and magnesium and how to keep the bones strong but not calcify the soft tissue. And that's Wednesday, February 15th at we don't have the, it's February 25th. We don't have the title of the webinar yet. So once again, thank you all for coming. Thank you, Patrick, for presenting to us today. And that concludes the webinar.